Welcome everyone, and it's my great pleasure to introduce today's keynote speaker, Yann Hautier from how do I spell it? Utrecht University. Yep. And Yann went to Lausanne University Lausanne for his master's and also his bachelor's, and then he did his Ph.D. at University Zurich with Andy Hautier. And he actually had moved to University of Oxford. And Yann is assistant professor at Utrecht University since 2015, right?
That's right, exactly.
Okay. And he's well known for his work on biodiversity, so we share some common interest on the causes and the consequences of biodiversity. And I really like his work on the published in Science and in 2009, demonstrating the importance of light limitation for plant coexistence. I think that work belongs to every ecology textbook. And most recently he's been working on the effects of anthropogenic environmental changes on biodiversity and stability. I think today he's going to be talking to us about some of his recent work. Yann?
Yes, well, that's my pleasure to be here while I'm home, but here with you, virtually with you. Yeah, thanks for the introduction, and thank you so much for the invitation. I'm very happy to be able to share my work, and I'm going to actually present some novel results that are unpublished that should be coming available later this year. So yeah, I'm a plant ecologist, and I'm very much interested into understanding the impact of the loss of plant diversity on the functioning of our ecosystems. And the ecosystem I'm working mainly is grasslands. Of course, I'm interested into other ecosystems as well, but that's the one I'm working mainly. And then I'm interested into different aspects of the functioning of these grasslands, but particularly their stability. And so that's what I'm going to present today. I'm going to present some work about links between biodiversity and stability. And I'm going to focus on natural system, and I will explain some of the differences. So this is what the presentation will be about. I'm going to make a little brief history about the diversity-stability relationship. I'm going to go through different mechanisms that are underlying this relationship. And I'm going to do that at the local scale, and then I'm going to scale up to larger spatial scale, showing some recent development and some recent evidence about this mechanism. And then I will present these new results that come from a review that will be published in a science book that should be published later this year. And I will also show some major challenges that we need to tackle in the future. The research, the link between biodiversity and stability has been a very long history. More than a century ago, this quest has started. And with some very famous people like Odum, Mark Hautier, or Elton already in the 50s. And this has been a long search trying to understand what makes systems more stable, and if biodiversity is indeed one of the reasons why systems are more stable. And in the 50s, this was strongly believed, based on observation and intuitive arguments, that diverse and complex systems are more stable than simple systems. And these observational studies were actually lacking some theoretical and experimental foundation. And in the 70s came a new paradigm. Actually, that's the reverse, that diversity and complexity begets instability instead of stability. And these were results that came from strong mathematical rigor and theory. And therefore, this really helped led to have this new paradigm. So we have two different paradigms here. Diversity begets stability in the 50s, and then diversity begets instability in the 70s. Of course, there are a lot of limitations into these studies, but the main ones is that they differ into the definition of stability and the different scales of stability at which this series and these observations were carried. And you can see it here because biodiversity and stability are both multifaceted concepts. So I'm going to try to put the pointer. There's like a laser pointer that I'm going to try to find out. Here it is. So biodiversity has multiple aspects. You have local diversity, richness, evenness. We have functional diversity, phylogenetic diversity. Of course, also larger spatial scale diversity like community composition or beta diversity. And stability also has multiple definitions that can be applied at different ecological organization, species or population versus community or ecosystem level. And then you can measure stability in different ways. You can measure temporal stability, which is a kind of longer term measure. And you can also measure shorter term measures of stability, which are resistance, resilience, and recovery. So all of this diversity, this multifaceting of these different concepts has made the whole quest quite blurry. And what I would like to do here is to show several facets of diversity, and this will allow me to actually make the definition of that. So here you can see a system where you have two species, a yellow species and a blue species, and they are varying through time. Okay, and these two species, when you sum their biomass, then you have this black line here. Okay, so that's the biomass, abundance, or cover, whatever it is. I'm just going to talk about biomass, and that's biomass fluctuation for each species, and the sum leads to this black line. And you can measure the invariability, which is the inverse of the coefficient of variation, which is given by the standard deviation divided by the mean, and then you have the stability, which is the inverse. So you take the mean divided by the standard deviation. So it's the temporal mean, the temporal average production, divided by its deviation, the standard deviation, so its variability. And here you can have an aspect about the temporal stability. And of course, longer term data gives longer temporal stability. And then you can see that you can measure stability at different organization level, because you can measure the stability of a community, which is this black line here, which is given by these two species. That's the community. But you can also measure a species level stability. And that's one of the big argument that there were in this previous controversy in these findings, is that these were applied at different spatial scale, organization level. And then you have, so that was for the temporal stability, and then you can take this temporal stability and make it into shorter term parts, and then you can measure resistance to a drought event, for example, in this case. So you can measure the production of biomass at a certain time, and then how it changes during the drought event, and also how it recovers after that. And therefore, after all, you can measure the resilience of the system, so how it comes back or close to the original state. So that's the different kind of stabilities I will mention. So I will focus here mostly on the temporal stability, and I will do that for one function, which is biomass production. So that's what we will be looking mainly. And then after these 50s and 70s publications and paradigms, came studies in the 90s, the first experimental studies. But you can see here, plant diversity, so plant richness, was not manipulated. But in the studies, and in the studies in the 90s, led to two seminal papers, and one of them is this one, and the two were led by Dave Tillman. And in this one, they look at the link between plant species richness and, in this case, resistance to drought on the left side, and resilience to after drought. And you can see that when you have a higher richness, you have a higher resilience resistance and a higher resilience. And so that was one of the major findings of these studies. And similarly, but looking at longer term data, so the stability. Okay, so this is the coefficient of variation. This means that lower values means more stable. Okay, so you have to put it the other way around. And you can see that in this case, when you look at the community, so this is community biomass, you can see that when you have a higher richness, you have a higher stability at the community level. But the interesting findings of these studies is that they also look at the population or the species level stability. And you can see that higher diversity means higher variability, lower stability of the constituent species. And so this finding that the stability can take different direction of effects depending on the level of organization really helped to understand the earlier difference in paradigms. However, these studies were highly criticized, both of them, by Houston in 1997, that explained that the levels of diversity that are in this system are actually not natural gradients, but instead the level of diversity is due to the fact that this is a fertilization experiment. So the fertilization created these gradients in diversity. But the problem is that when you look at the relationship between diversity and stability, then fertilization impacts both diversity and stability. And therefore, you cannot argue that this relationship is a causal one. And therefore, there might be many different covariates that act to explain this relationship between biodiversity and stability. And I can show you this here. So when you have a global environmental gradient or change, for example, fertilization, this fertilization will have an impact on biodiversity, and it will also have an impact on stability. And then in this study, what they do is to look at then the resulting biodiversity-stability relationship, which is actually caused by the fertilization. So to show you this, I have this little figure here, and you can see here, for example, this is diversity and stability, species richness and temporal stability. And you can see in ambient condition, this is this blue date points here. And then you have this red, which is when you have the fertilization experiment, for example. So when you fertilize and when the response is in opposing direction, for example, when you fertilize, you have a reduction of richness, but an increase in stability. Then the resulting relationship will be negative. But this negative relationship across these treatments are not a causal relationship. They are the result of the fact that fertilization reduced diversity and increased stability at the same time, this opposite effect. If the effects are in the same direction, so when you fertilize, you decrease diversity and you decrease stability at the same time, then you can see that the resulting relationship is positive. And then it's null if one of the two diversity or stability does not respond to the treatment. And this has caused really people to think about these problems of these experiments that create this gradient in diversity by a treatment and then looking at the resulting relationship. But of course, Dave Tillman thought about it earlier, and he created this, he set up this experiment, which is called Big Bio. It's a biodiversity experiment where they manipulate plant species richness, and then they look at the impact of this on stability. And in 2006, Dave Tillman came with this paper showing the exact same result that he had shown 10 years ago in the 1996 paper, that stability is higher with higher diversity at the community level, but at the species level, we have the reverse happening. So again, showing this difference, difference in direction of the effect of species richness on stability based on the organizational level. And this helped to reconcile the earlier paradigms. This was confirmed later by a study led by my former supervisor, where we showed that using the BioLabs data, which is a network of biodiversity experiments, you can see we had 8 sites in Europe that were manipulating plant diversity with different levels, and we found the exact same result, that there is a higher stability, lower CV, So higher stability at the community level when you have a higher species richness, and the reverse for the species level. Well, after many experiments that have looked at the relationship between plant diversity and stability, then we have come to the consensus that indeed plant diversity, at least at the community level, leads to higher stability. Okay, so that's what the consensus is currently, and you will see. I will show you that most of this knowledge come from biodiversity experiments. So experiments really manipulating diversity. That's the only way we can have a causal relationship. And then the next quest has been to look at the mechanism underlying this diversity stability relationship. And there has been several that have been advanced. Overyielding is one of them. Overyielding is simply the mean here in the equation. Okay, so if you have a higher temporal mean, for example, if diversity promotes productivity, then you have a higher productivity. And if this productivity, this increase in productivity is more than the increase in the variation that goes with that, then you tend to have this overyielding effect where you lead to higher stability. So simply increasing the productivity more relative to the standard deviation gives you this stability effect. And you can see it here. This is the BIDEPS experiment once again, but this is the community level data. And I look the stability here, and you can see that you have higher diversity, higher stability. And if you decompose this aspect into here, this is the mean, the temporal mean, and this is the temporal standard deviation. So if you decompose this stability into these two components, the mean and the standard deviation, you can see that the mean increases faster than the standard deviation, and this is what leads to this higher stability. So overyielding is indeed part of the aspect. I wanted to see whether this was true also in natural system, and so what I did is I looked at the data from the neutron network. The neutron network is actually an experiment where we manipulate fertilization, N, P, K in all the different factorial combinations, and there is also a fencing treatment that is crossed with the NPK. And within this, and this study is replicated around the world, and in a way that we have standardized protocols so we can really put all this data together. And I could use the control plots from these systems to actually look at the gradient of richness and if this was linked to higher stability, as we find in biodiversity experiments. And this is what I did, and this is the result. So this is the BIDEPS experiment. This is exactly the same figure as I showed you before. Okay, so you have the stability here, and you have the mean and the standard deviation. And here, this is the neutron network. Okay, and now you see this dashed line here. This dashed line is actually reporting the results from the neutron network. Okay, so the dashed line is this exact line here. You can see first, which is pretty impressive, is that these relationships are very similar in biodiversity experiments as to natural systems. So that's the first very impressive result that we find here. But what was, I think, more interesting is that when you look at the mean to standard deviation, you can see actually that the mean doesn't change much. Okay, so you don't have this kind of overyielding effect. But instead, you have what I call the real stabilization, because the standard deviation is decreasing. Okay, so this is really for me the stabilization because the variation is decreasing. In the BIDEPS experiment, you can see that it's mainly due to a higher productivity, with higher diversity, not really that you have a decrease in the variance. You have a decrease in the variance which is proportionally lower. Okay, so the increase in the variance which is proportionally lower, and therefore this is leading to a higher stability. But here we have a real stabilization effect. So overyielding is certainly acting, but in natural system we have actually more stabilizing effect via a decrease in the standard deviation. Another mechanism that has been proposed is species asynchrony. And you can imagine the system that I showed you where you have two species that are varying through time. And when you sum these two species, you have the community biomass. And of course, if these two species are acting in the same direction, when a year is good for one species, is good for the other, then productivity is very high. when it's low for one species and also low for the other species, then the productivity is low, and therefore you have huge variation. And if this is the case also with higher diversity, if all the species are responding in synchrony, they are not asynchronized, but they are responding in synchrony, when a year is good for one species is good for the other species, then you can see that at the community level, this is leading to a huge variation as well. So how can we stabilize this system? Well, we can stabilize this system if the plant species are responding in asynchrony. This means when a year is good for one species, it's bad for the other, and when it's bad for one species, it's good for the other. Therefore, the species are able to compensate in their biomass through time. And this compensation then stabilizes the community level biomass. And I'm showing this that this can happen if you have only two species. You don't need many, many species. If you just have two species that are in perfect asynchrony, you will stabilize the system. But of course, what happens is that when you have a high diversity, then you increase the likelihood, the chances that at least one of the species that is there will really compensate for the decrease in the other and therefore stabilize biomass. And what's the evidence for that? So the evidence comes also from biodiversity experiments. This is again the BioDeps network of biodiversity experiments. This is the stability relationship that you've seen with richness. And now this is the asynchrony. And you can see that in this study, indeed, when we have higher diversity, we have higher asynchrony. And this is also one of the mechanisms that really contribute to stabilize biomass. And now this is the BioDeps here on the right, and on the left you have again the neutron network control the ambient plots. And you can see again this is the same relationship you've seen before. You can see again that we have this asynchrony there that is contributing to stability, and you can see again that it's very, very close to what we have in biodiversity experiments. So again, a pretty impressive results to see that this is also happening in natural system. And there is more and more evidence on that. There is a recent paper by Valencia here that showed this strong link between asynchrony and stability. So if you put an A instead of synchrony here, then you take the relationship in the other direction and you have higher asynchrony, higher stability. Okay, so asynchrony is really a strong mechanism leading to stability. Another mechanism that can happen is actually species stability. So species stability. So if the constituent species are more stable, which is happening here in this case, as compared to here, then again you can stabilize the whole system because the constituent species are less variable and therefore the contribution to stability will be less variable and therefore the community stability will be more stable. Okay, community biomass will be more stable. So that's also one of the mechanisms by which we can have this stabilization happening at the community level. And there is some evidence that indeed this is happening, that higher population stability leads to higher community stability. Now I've been showing these results and they are based on local scale. So basically communities. Okay, so take a community in a plot that we say this is a plot of that size, 1 square meter, 5 square meter, whatever. And we look at this local diversity, species richness, and the local stability, which is the community level. How do we scale up our knowledge? And why is it important? It's important to scale up this knowledge. Why? Because there is a mismatch between the biodiversity experiments or the understanding that we have how biodiversity is changing ecosystem environment and scale at which biodiversity is being lost, which is at the larger spatial scale. And there is, of course, big debate now to know whether we are losing plant diversity at the local scale. But there is a mismatch between this knowledge. And so we have to scale up this understanding that comes from biodiversity experiments and observational studies to higher spatial scale, temporal, but also, of course, spatial. And this is what we need to do. We need to go from this scale, these very small plots, to a regional scale or even global scale. And there has been some development in the series recently that have been really allowing to scale up this understanding. And this is based on a paper from Xiaopeng Wang and Michel Laurent in 2014. And this paper came out, 7 years ago now. I was really fascinated by this paper, although I could not understand everything. But then I had to write a paper about it because I was so excited about it that I digged into all the potential mechanisms. It's a theoretical paper, so it's pretty, full of mathematical equations. But I'm going to summarize here for you what's happening. What this figure is showing you is basically exactly what I've been showing you before. The species stability, the species asynchrony, and alpha stability is basically the community stability. So here, this dark green line is the community levels, what's called the alpha stability. And then here you have the constituent species. So if you have high variability in the constituent species and low asynchrony, then you can see that basically you have a low stability at the community level. If now you have high species stability, this contributes to stabilized community level. Or if you have high species asynchrony, then this also contributes to community level stability. Okay, so that's what I've been showing you before, and what Xiaopeng has been able to do is to actually decompose this stability here, the community level stability, into these two constituents, the species and the species stability and species asynchrony. But he has been going a step further. This is why we have the scaling up. And the scaling up has been basically to apply exactly what we have here at a higher spatial scale. And you can even imagine that you can go into much higher spatial scale. And the gamma stability is then defined as the meta communities. Okay? And so you have these meta communities, which are multiple communities in the landscape. And of course, if you have a higher stability of the constituent communities, then you have a higher stability at the gamma scale, at the next level. And then if you have also these communities that are responding in asynchrony, so if the biomass of these communities are responding in asynchrony, then these can also contribute to spatial stability, to stability at the gamma scale, as species asynchrony contributes to stability at the alpha scale. So that's exactly the same kind of principle. And he has been working on this mathematical decomposition, linking these different things. So you can see here that in this case, here on the right, these two communities are responding in asynchrony, and this is what really stabilizes the meta community, the gamma scale stability. Okay. So do we have evidence that this mechanism, this, of course, species stability, species asynchrony, but also spatial asynchrony is working? And there has been a paper from Kevin Wilcox in 2017 that has been showing that indeed there are strong links between the constituents of alpha stability, species stability, and species asynchrony on gamma stability, but also that spatial asynchrony was playing a strong role in this system. What is interesting actually is that they didn't really find any link between diversity and any of these constituents. They found a link between species richness and species asynchrony here. So higher species richness, higher asynchrony, therefore contributing to higher stability at both alpha and gamma scale. But they didn't find any link between beta diversity, so the dissimilarity in species composition among the communities, and how these can help to stabilize, to make the communities more asynchrone, and therefore to contribute to stability at the larger spatial scale. But I also did my looking at little studies looking at this using the neutral network data. So this is again looking at the ambient, the control plot from the neutral network. And we found strong link between species stability, species asynchrony, and alpha stability. Strong link between richness and asynchrony, as Kevin Wilcox did. But we also actually found this link between beta diversity and spatial asynchrony. Okay, so we found that indeed beta diversity, so this dissimilarity in species composition among communities, is important for asynchrony among communities, and therefore for stability of the higher levels. And why this result is important? It's important because it means that if we think about the way we cultivate crops, if we do these kind of things on the right, which is having this monoculture in huge, occupying huge space in the landscape, then we have low beta diversity. Okay, if you can think about a system like this that has just one species, it's basically no beta diversity, so no diversity in the landscape, as compared to a system like this that has a high beta diversity where each different field is a different crop species. And therefore, this result suggests that if we diversify our landscape, the crops within the landscape, of course, a crop is still a monoculture, but if we put different species in monoculture, we increase beta diversity, and therefore we can increase the asynchrony in response to environmental fluctuation and maximize, let's say, stabilize food production at higher spatial scale, which can be regional or national or even global spatial scale. So that's very important consequences for that. Now that I've showed you the different mechanisms and how they play at different spatial scale, then I can touch on this, the result of this review that we are going to publish hopefully soon this year, that look at the mechanism, the strength of evidence, the balance of evidence between that leads to the mechanism and the link between diversity, asynchrony and stability at multiple spatial scale. This is basically the framework that we are using. So we're looking at diversity, the richness, and composition, so dissimilarity in species composition among communities, and how this can help to stabilize the biomass production through asynchrony, overyielding, so species asynchrony, but also spatial asynchrony, and then how this contributes to stability at the local and larger spatial scale. So we went to make a review out of this. So we looked at studies. We had 39 studies here, and you can see that these studies are covering natural gradients, so really looking at just natural gradient of diversity and how this relates to stability, but also studies that actually manipulate a global change driver, fertilization, grazing, or multifactorial experiments that look at how these global change drivers impact biodiversity and stability, and therefore the biodiversity-stability relationship. And you can see that this covers a lot of drylands and deserts and temperate grasslands, but many ecosystems are missing, or let's say very much underrepresented in this study, in these studies. And terrestrial plants are the majority, and you can see that for the rest, we don't have much information. Okay? And we cover just natural systems, for whatever natural can be. So semi-natural system where we have a natural gradient, or we have an experiment that manipulates a global change driver that influence both diversity and stability. And then we looked at the strengths of evidence for the mechanism behind the diversity-stability relationship. And I'm going to start first with the results from the natural gradient. Okay, so how this is happening in natural system, natural, semi-natural system. And these are the results. Basically, what we found is a very strong link between diversity and species asynchrony, overyielding, and therefore this mechanism contributes strongly to stability at the local and larger spatial scale. Okay, so this was pretty striking, very important result that we found in this observational study, so in natural systems. A few studies only found a negative relationship between diversity and species asynchrony, but you can see the majority found positive results. And then the strong contribution of local diversity to the mechanism driving to stability at the local and larger spatial scale. But we also can see that diversity at larger spatial scale, beta diversity, also contributes strongly to spatial asynchrony, and that this is also contributing strongly to stabilize biomass production at higher spatial scale. Now that I looked at we looked at the balance of evidence in these natural systems, we can look also at experiments that manipulate one of the driver and look at the impact of this driver on the diversity and stability, but also at the diversity-stability relationship. So there are two aspects here, and I want to make this clear. So one aspect is when you add a global change driver, fertilization, grazing, then as I told you, you change biodiversity and you change stability at the same time. And again, if you reduce one and if you have opposite direction here, reducing diversity while at the same time increasing stability, then you have a negative relationship. If you have both direction, decreasing both diversity and stability, then you have a positive relationship that goes out of it. So these individual effects, which are direct effects, these are causal effects here on diversity and stability, lead to a non-causal relationship here between biodiversity and stability. Okay, and many people have looked at this, and we want to here show the strength of evidence for that. Another aspect is studies that look at the impact of this global change driver on the biodiversity stability relationship here. Okay, so how this relationship is changing in response to the global change driver, and this is a causal link because it's really looking at how this changes the relationship. And this is the change, for example, here from this blue line to this red line. You can see that the relationship here changes from positive to neutral. Okay, here also from positive to neutral, from positive to neutral. And of course, you can imagine the reverse, where the relationship changes from negative to positive. Or you can imagine all the scenarios here. Okay, so these are two aspects. One aspect is this black line, which is the relationship across these studies that manipulate a global change driver and had a causal effect on diversity and stability, but not on the diversity-stability relationship. And the other is looking at, so that's the black line, and the other is looking at the change in these relationships in both systems. And this is summarized here, and you can see here, biodiversity and stability means joint effects on both biodiversity and stability. So that's these two arrows here that leads to a relationship that is either negative, neutral, or positive. And when you look at the strengths of evidence for these different studies that either look at fertilization, grazing, warming, or drought, you can see that for fertilization, there is strong evidence that actually in this system, when you fertilize, you lead to a similar effect on both diversity and stability, which is usually a decrease in both, a decrease in diversity and at the same time a decrease in stability, leading to a positive diversity-stability relationship. Of course, some relationships are neutral and very few, one only, is negative. For grazing, it's mixed evidence. For warming, pretty much neutral evidence, and then again, pretty neutral evidence for drought experiment. Now, if you look at the impact of these drivers on the biodiversity stability relationship, which is this path here, which is the change from this blue line to this red line here, then you can see that fertilization, we have only two studies, but these two studies go in the same direction. That's when you fertilize, you reduce the diversity stability relationship, so going from positive to neutral. And then in grazing system, warming or drought system, we have very few studies, and they are not convincing in their direction. Now I want to show you, go a little bit deeper in that, because I want to show you the results of the nutrient network. I showed you before the result from the unmanipulated, the control studies, and you can see here we have this positive relationship between diversity and stability, and this is asynchrony and how they contribute to that. You can see that this positive relationship in fertilization, in fertilized field, so this is again reporting this line here, the dashed line, and then this is the relationship in the fertilized communities. And you can see that in these communities, the relationship changes from positive to neutral, so the relationship is weakened. Okay, so fertilization weakens the the stabilizing effect of biodiversity. And we can see that this is through a reduction in the asynchrony mechanism. So when you reduce asynchrony, you reduce stability. So strong evidence for that. But this is only one study, or a big study with many, many different sites. But we have also some evidence from different papers, also from your lab. Then what I was interested into looking at is, again, does this scale up? Does this effect that we see scale up? And this is looking at now, this is the control, and I showed you the results from that before. Okay, so that's the ambient, and that's the fertilized plot. And what you can see is that every of this link that we have here between diversity and the mechanism contributing to stability at the local or larger spatial scale, all of these links are reduced with fertilization. So the neutral link here becomes negative. The positive link here is still positive, but weaker. And this positive link becomes neutral here. So fertilization generally leads to a lower stability via decreasing the contribution of diversity at multiple spatial scale to stability. And that's again a very important output from this, and I explain why it is important to look at this diversity in the landscape and how fertilization is impacting this has very strong repercussions, and we have to be very careful into fertilizing, or at least all the damage that fertilization can cause to these grasslands. And then there was this very important study also, and I want to compare this with that. Why? Because in this study, which is also looking at nitrogen enrichment, this is again the result from your lab, you can see that fertilization reduces also diversity and therefore stability at a local and larger spatial scale. You can see that fertilization also reduced beta diversity, but actually there was no link between beta diversity and spatial asynchrony. So this link was not found, and therefore spatial asynchrony can really be a stabilizing effect that is still strong even under fertilization. We didn't find these results, although let's say the approach of the study is quite different, because again we look at the relationship, at the impact of fertilization on the relationship, and not the cascading effects of nitrogen enrichment on different spatial scale. But also very interesting result. This was, of course, one site. The nutrient network has many, many different sites. So, but it's nice to see that some of these are similar, and of course, some of these can be different depending on the site. Now, I just want to cover some future challenges that we identified when looking at this. One of the very important aspects, I think, is that many of these ecosystems are understudied. We've seen that temperate grasslands or drylands are the most studied, but we miss information on ocean, tropical rainforests, etc. So many are understudied, and these ecosystems are also of high contribution to the stability. The stability of the system is very important in many aspects, and therefore neglecting to understand how the systems are working is really hindering our understanding of how this system can be more stable in the future or more affected by global drivers. Of course, the stability of biomass production has been more studied. We know so few about other ecosystem functions, carbon storage, pollination, etc. And of course, the trade-offs between these functions. So how this is working? How is, biomass production is easy because it's easy to measure year after year, and there is strong theoretical background on it. But we need the theoretical background also for other functions and to study them. Most studies look at species richness. It's again it's easy to measure, and some studies now are looking at composition. But we need studies that are looking, for example, at functional diversity, so how the traits contribute to the stability, to really understand why a species respond like this and which traits are corresponding to that. Can we link this, for example, to the leaf economic spectrum and have this understanding along this spectrum, which is a very general spectrum that can be applied at multiple dimension, multiple spatial scale, and multiple ecosystem types, etc. So that we can have a general prediction of how this, based on these traits, how this ecosystem will be changed in the future. And then we need to understand the stability of the whole ecosystem. So not only we need to measure different functions and to look at their stability, but we also need to have this broader aspect that looks at multiple functions at the same time, and therefore multifunctional stability. And again, understand the trade-offs between functions. You cannot maximize all the functions at the same time. This is just impossible. So what are the trade-offs there? Can we maximize, stabilize biomass production, but what are the functions that will be trading off with that? So we also need to understand this. And then we need, of course, to understand the link between stability, diversity and stability of ecosystem services, so not only biomass production, but again, higher scale of services. And I think that's it. I'm done with my presentation. So yeah, I would like to thank a lot of people here. I don't have a special slide for that, but especially Fons van der Plas, which with whom I've been writing this book chapter. The results I've been showing from this review come from a collaboration with him. And of course, a lot of the data come from the Neutral Network. So I would like to thank all these people here. And you've seen also some results from the biolabs experiments. So Andy Hector and all the contributors of this biolabs experiment have to be acknowledged here. And yeah, thanks for listening. And I'm very happy to take any question. I think I'm quite on time, actually.
Right on time. Thank you so much, Yann. Great talk. Any questions for Yann? The audience? Actually, I have a question, and it seems like if you look at the effects of anthropogenic environmental changes on stability, and it changes both. It would affect both stability and biodiversity, and so you have to statistically tease out the direct effect from on stability, and also its indirect effect through changing biodiversity. So I was wondering have you ever thought about any experiment controlling both biodiversity and environmental changes independently?
Well, that will be pretty hard to do, I think.
Yeah.
I never thought about it, actually, but that sounds like a logical next step. The question is, can it be done?
Yeah, because when you for example, down nutrient into grassland, it typically change richness, plant richness, right? And suppose it doesn't change richness, then you should be able to tease out the direct and indirect effects, right?
Exact. So we've been trying to do something a bit similar in the Neutral Network. And maybe I can show if I have this here. Do I have this here? Yes, actually I have it. So maybe I'll share my screen again. Here. Okay. So it's not exactly tackling this. So my answer would not be satisfying, I think, at this level. But what we've been looking at is, I told you in the Neutral Network experiment, we look at how fertilization impacts the diversity-stability relationship. And we can see that fertilization takes this positive relationship that we have in natural system and makes it weaker to neutral, basically. And one of the obvious reasons why this might happen, one obvious approach is that there is just a direct effect of fertilization that is due to nothing else than fertilization. Another aspect is that you can decrease species asynchrony, and this is one of the mechanisms. We've seen that this is playing part. But another aspect is that it can be also that you have a diversity-linked effect, which is basically you fertilize, you lose diversity, because there is a positive diversity-stability relationship. By losing diversity, you decrease the stability of the system. Okay, and what we see in the nutrient network is actually this flat relationship. So we don't have this relationship between richness or beta diversity with change in alpha or gamma stability. So in other words, the impact that we see in the nutrient network, the reason why this diversity stability relationship is weakened is not due to the fact that we are losing plant diversity. And that's quite interesting. And I was a bit puzzled by this. Also because we actually found strong evidence, and I don't know if I have this here. Maybe not. Yeah, okay. I don't have this here. Because we found strong evidence, yeah, from this one here. We found strong evidence that this is the case when we look at this data, which is data from Cedar Creek. And you can see here, we have many different global change drivers. So nitrogen addition, carbon dioxide, fire, herbivory, water. And we also have the two biodiversity experiments that are running there, which is the Big Bio and the Biocon. And they are summarized here by this red line. So you can see that you have a strong diversity stability relationship. So if you lose species, you lose stability, okay? And we found exactly the same in this natural system where you apply some global change drivers. And so based on this tells you that if any global change driver that leads to a reduction of plant diversity will in turn lead to a lower stability. And the funny thing is we don't find this in the nutrient network. And I'm quite puzzled about this, and I don't know why this is the case. And it could simply be that we have too short amount of data to actually look at this, so that's these effects take longer to appear. When you look at the Cedar Creek studies, some of these studies are running for more than 20 years. So we have much longer data sets, longer framed data sets. So that could be one of the reasons. Maybe the impact, the decrease in diversity is too low in nutrient network as compared to other systems to really have this diversity-driven negative effect. So I know I'm not answering exactly your question here, but this is the way we try to disentangle these different effects. Now, of course, the best would be to really manipulate, to have the manipulation experiment that really tries to manipulate both diversity and functioning at the same time. Let's say, to really disentangle these two at the same time. But that would be the hell of a job, I think.
Yeah, I know it would be difficult because when you manipulate the environmental factor, typically would influence species richness.
Exact. Yeah. It's very hard, for example, to keep a certain level of diversity when you fertilize.
Yeah, that's what I'm saying.
I mean,
yeah,
you have
exactly.
really hard. Of course, you could try to make a system to reach the same level as the fertilized system. I'm not sure, but yeah.
Maybe low level of species, just few.
Yeah.
Any other question for Yann?
Yeah, hi. This is Mike Gittesman. I'm not. This is not really my area, and I know you've been speculating about this, but I'm having trouble. Again, the mechanism between fertilization and stability. I don't get it. You probably went over it many times, but what is it again? Why is that happening?
One of the things is maybe I can come back to some slides. So one of the slides I want to show is this one here. So one of the mechanisms that really contributes to higher stability at higher diversity is this asynchrony in species responses. In response to environmental fluctuations. So you have these species that fluctuate, and if they respond in synchrony, so if they are all showing the same response, good for one species when the year is good, and bad for the other species. Sorry, good for the two species, or the species when it's good, bad for the species when it's bad. Then they respond in synchrony, and there is no compensation possible. Okay, and you can see that when you have a higher diversity, you have a higher likelihood to basically have some species that are well suited for the environmental condition that is there. And so if it's a very dry year, of course you have a lot of species that will be suffering from that. But there are some species that are being released from the competition for other species, and that will thrive under these conditions. And they can compensate for the loss of functioning of the other species and therefore stabilize the system. And fertilization acts on one of those. Here you can see that species asynchrony is reduced at higher diversity level. And how this is happening, then this is the question, why this is happening. What makes that when you fertilize, you synchronize the communities in response to environmental fluctuation? And there could be reasons for that that are based on the traits of these species. So for example, one of the things that can happen is that you have, when you fertilize, you normally drive the system, the community composition, so you lose species, that's one thing. But usually you also drive the community composition towards species that are these fast species that are able to extract these nutrients really quickly and grow really quickly. And these species are, this fertilization there might be that it's driving this system to be really on this fast track, and that is responding in the same way because you have less species, but also because they are more synchronized in the way they do because you drive the system towards these same species that are responding the same way. Now, on the other hand, I would argue that this might be also a counter-argument because in the end, these species that are on the fast spectrum are also supposed to be less resistant against, for example, drought and so on. And so maybe this could also drive the system to be less stable overall because of this lower resistance against, for example, drought or other system. So there are some counteracting forces here. But I don't have the answer in the detail of exactly how this is happening. And we are working on this. We are working on getting traits data, which is not an easy thing, especially when you need to find traits data for more than 2,000 species. And then to link this to these responses to see whether we see some of these things that I'm explaining here. So we are working on it, but for now it's more like bigger, let's say, patterns that we see. But we're trying to dig deeper into it.
Okay, thanks for that.
Any other questions? If not, let's thank Yann again for a great talk.
Yeah, well, thanks again for the invitation.